let's get started. So I did post lecture one. If there's anything that you feel like you need to go back and look at again, or if you weren't able to come, there's a tab on Blackboard where you can find those. You can be working on the chapter 1 homework. We finished chapter 1 last time, went through some history, some kind of basics about what microbiologists study. We're going to continue talking about culturing organisms a little bit today and get into microscopy, a little bit of taxonomy. I wasn't very clear. I actually, your first homework assignment is just kind of a, hey, let's get started, make sure you're logged in kind of assignment. Some of you have done that, but I have that set to be due next Friday. So all the other homework assignments are due the day of our first exam, but I really want you to make sure everything's working, you get the code, you got logged in right, and probably about 2/3 of you have done that so far. That's something to work on this weekend, but for sure if you haven't done that yet. So if you are having a problem, you know, whatever, your financial aid doesn't come in or something like that, just let me know and I can extend that first assignment, but try to make sure that that is working for you. Okay, so we do have class Tuesday. Just, you know, if you're in lab, there's no labs on Tuesday. Just want to remind you that we will be here again on Tuesday. Okay, any questions from last time? Okay, don't be afraid to ask. Stop me anytime. If you ever can't hear me, make sure you let me know. But where we let off last time, we were talking about growing up organisms and some of the terminology that we use for that. There should be a little overlap if you're in lab this week. I said some of the same things this morning. But where we left off, we were talking about solid media. And remember, I think I mentioned that solid media contains agar. Okay, and I think that I got through that agar is really just a polysaccharide that we get from a special kind of seaweed. All right, so what's special about agar, what makes it a really good solidifying agent, is that bacteria cannot metabolize it. Okay, so every once in a while I hear maybe, you know, on the news or something that they're talking about growing bacteria, and they say bacteria eat the agar. Okay, that there is agar in the medium and bacteria eat the medium, but they don't eat the agar. Okay, if bacteria were able to eat the agar, then the media would liquefy, okay, because it's the agar that makes it solid. Okay, so we don't want, it took us a long time, we haven't found anything as good. We've been using agar since like the 1880s, and we haven't found anything that works better to keep microbial media solid. Okay, think about something like gelatin, which is also a solidifying agent. If you put Jello in an incubator that's like body temperature, it melts. Okay, if you've ever had gelatin at a picnic or something, it will melt. Okay, and in addition, bacteria, they can sometimes make an enzyme called gelatinase that digests gelatin. Okay, then that liquefies it too. All right, so the great thing about agar, there may be some exceptions somewhere, but the vast majority of bacteria are not going to digest it, so it stays solid, and even stays solid in the incubator. Okay, so we've been using that, like I said, for a long time. Haven't found anything better than that. Okay, and the really nice thing about solid media, I can't remember whether I said this or not, but it's easier, oops, that's an R, to keep cultures pure. Okay, so like I said, I forget whether I defined that or not. I've said this so often in lab this week that I forget what I talked about and what I didn't. But a pure culture is one where there is only one species of organism growing. And whether that's a plate like here where we put agar medium into a petri dish, or whether we put it into a test tube, we call that a slant. You see a slant of agar right here. It's allowed to harden, cool off kind of at an angle. The great thing about solid media is that we can kind of tell if we have one species growing or if we have many. Okay, if you've got organisms growing in a test tube, you know, everything pretty much looks alike, and it's hard to tell if you got one thing or lots of things. Where do we get to agar? We actually have to harvest it. Okay, it comes from red algae. We haven't been successful in farming red algae and just, you know, growing it, whatever, in a pool or whatever. We have to go and collect it after storms, places like Morocco. So that's where our agar comes from. Just kind of a FYI. Okay, so I was talking about pure cultures. Okay, usually that's our goal in the microbiology lab, to be working with one organism at a time. If you've got several mixed in there and you're trying to characterize that organism or identify it, you're going to get conflicting results. So usually that's our goal. And in order to know that our cultures are pure, we have to be able to see isolated colonies. So on this plate right here, here's some isolated colonies. Okay, remember, bacteria generally grow up in these discrete packets. Each colony is millions and millions of cells. Okay, when there's so many of them, then we can actually see them with our naked eye. Okay, so when those colonies aren't touching any other colony, we call that an isolated colony. Okay, so in order to get isolated colonies, we generally have to spread the cells out quite a bit. So ways that we do that, if you're in lab, you did a streak plate, one of many that you're going to do. This is actually the streak plate technique down here. You originally, looks like they started rubbing the cells on down here. You clean off your loop, you sterilize it, and you spread those cells out a little more. You clean off your loop, spread them out a little more, and you can see how the cells have gotten more and more dispersed till in the fourth quadrant here, you can only see a few cells after they grow up. So that's what we want. We can look at those isolated colonies and see that they all look almost the same, exactly the same, in fact. And so we can be reasonably sure that that's only one species on that entire plate, in that entire culture. Okay, over here where you can see there, the growth is kind of all together. It's hard to tell, you know, what those colonies look like there. Okay, so one method is the streak plates. You're also going to do a spread plate if you're in lab, and you would have done a pour plate today in lab. And I'll let your lab professor kind of talk more about those. They're just other ways to get isolated colonies. Okay, we want to keep our cultures pure. The way that we're going to do that is by using aseptic technique. So again, if you're in lab, that's what this week's lab was all about. Yesterday's and today's, practicing aseptic technique. And I'll let your lab professor talk more about that and harp on those things. But if you're not in lab, it's things like keeping the lid closed, flaming your loop, not putting caps down on the bench, wearing a lab coat, wiping your bench down with some kind of disinfectant. Okay, all examples of aseptic technique. Okay, here is a picture of a mixed culture. Okay, so a culture that's not pure. There is at least 12 different organisms growing here. You can see those colonies can take on a vastly different appearance, different color, different shape, different size, different sheen. Okay, so this is what we would call a mixed culture. Lots of different things growing there. You've got some isolated colonies, but definitely a mixed culture, right? If we wanted to identify what each one of those colonies were, we would need to touch a little bit of them with our loop, streak that out in a whole new culture, get it by itself, and then we could do tests to identify it. So Okay. All right, so a little bit more, I talked kind of basically about media, liquid versus solid, advantages, disadvantages. We have some more terminology that we use to describe media. There's lots of different microbial medias. It depends on what you're trying to grow up, really, as to what kind of media that you're going to choose. But that kind of, there's some, general principles that we think about when we're trying to choose a media. So I want you to be a little familiar with that terminology. All the media that we use is either considered defined or complex. So if it's defined, then the exact chemical composition is known. And complex is just the opposite. We don't know exactly what's in there. So exact chemical composition is not known. Okay, so that's a principle. Let me give you some examples of each kind of media. Don't memorize the examples or memorize the recipes. I'm just trying to use them to illustrate the principle. Okay, so here's an example of a defined media. This is from your textbook. It's also known as a synthetic media. Defined, synthetic, those two terms can be used the same way. It calls this a kind of media to culture E. coli. Honestly, I'm not sure why you would use this media to culture E. coli when there's a lot of all-purpose medias that E. coli grows really well on. But anyway, if you take a look at the ingredients of this media, we've got basically chemical formulas here. Glucose, we know exactly what this is, C6H12O6, right? So we know exactly what element in what amounts is in this media. So it's defined. Another example of a defined media is oftentimes using a defined media is sort of unnecessarily cumbersome, troublesome to prepare this kind of media because it's so kind of exact. But sometimes we use that media if we're trying to grow an organism that doesn't have very many requirements. And so you might only give it like a few things, and then organisms that need a lot more nutrients are not going to be able to grow. So if you want to grow an organism, say, that fixes its own nitrogen, you grow it in a defined medium that doesn't have nitrogen in it, and then only those organisms that you're looking for are going to grow. Okay, but again, that's just an example of a defined media. Here's an example of a complex media. And this is a media that if you're in lab, you're going to be using all the time. It's called nutrient agar. It's a good all-purpose media. I wanted to look a little bit more closely at what's in that media. It starts to give us an idea of the sorts of things that bacteria need to grow and reproduce. Peptone, basically that's just some short chains of amino acids. Okay, so makes sense in order for bacteria to make their own proteins, we have to give them some amino acids. A lot of bacteria can make up at least some, they can biosynthesize their own, at least some of them amino acids, but if we give them some in the medium, sometimes they need that. This next ingredient is what makes it complex. Beef extract or yeast extract. This is, say, beef that's been digested with enzymes to release a slew of nutrients. So things like carbohydrates and vitamins and minerals. Okay, so basically digested carbohydrates, vitamins, minerals. It's just a whole slew of things. We don't know exactly how much. Okay, so that's what makes a complex media complex. It has some kind of extract in it. It might be soy extract, it might be yeast extract. Okay, something that's just, had all the nutrients released from it. If it has agar, then you know that it's a solid media. Okay, nutrient agar. Okay, they're not eating the agar, they're eating all these, carbs and proteins and other things. And then a little bit of salt for osmotic balance. We're going to review osmosis next week, just to make sure everybody's clear on that. But we want to make sure the medium's maybe pretty close to isotonic. If you don't put enough solutes in there, then the bacterial cells are going to lose water, and that's bad when cells dehydrate. Okay, they're not going to reproduce and grow if they are dehydrating. Okay, so you buy that in a powder, you add water, appropriate amount of water, you adjust the pH and sterilize it, and then you pour it into, say, a plate or a test tube or whatever you want. So, okay, that's an example of a complex media. Again, it's going to have some kind of extract in it. Okay. Culture media can also be selective or differential. And you're going to talk about that a little bit later in lab, but we use several examples of both of those. Media can be considered one or the other, or both, or neither one. Okay. A selective media is a media where certain organisms are inhibited. Okay, there's something in the media that keeps some organisms from growing. Okay, it could be an antibiotic. It could be high salt concentration, and then only organisms that can tolerate a high salt concentration would be able to grow. Everything else would be inhibited. Okay, I'll give you some examples. An example in a minute. A differential medium is when certain organisms have a changed appearance. Okay, and that's going to be due to something in the media. It might be a pH indicator that will turn, some media or some organisms a certain color if they use a certain nutrient. Okay, it's really important to use differential media when we're trying to identify organisms, to characterize them. Okay, I'll come back to the anaerobic media in a second. Let me give you some examples. An example of selective media. Again, this is from your book. Here on your left, that's a nutrient agar. So that's the media that we just talked about. It looks like this sample maybe was a soil sample. It looks similar to that to me, where you would take a sample of soil, maybe add some water, mix it around, and then add the water onto some nutrient agar. You can see mixed culture, lots of different things growing there. Sabarod's agar is a selective media, and it has the same constitution as nutrient agar, with one big difference, and that it's pH'd a little differently. All right, so we've got a lower pH. Okay, so that acidic pH, actually many bacteria that would just normally be in soil are not able to grow at that low pH. Now, there are lots of bacteria that maybe are best suited for that, but sort of run-of-the-mill organisms here. Okay, this inhibited many of the bacteria. It actually favors the growth of fungi. Fungi tend to prefer a little bit lower pH. So you can see with the same sample what a difference there is in what's growing. Fungal, how do you tell if a colony is a fungi or a bacteria? You can't always, but fungal colonies are commonly bigger and really fuzzy. Okay, so you can definitely see some fuzzy colonies here on the right. There could be a little fungus over here, but again, the Sabarod's agar is selecting against a majority of bacteria because of that pH difference. Okay, so don't memorize anything about Sabarod's agar. It's just the principle of selective media. Remember, differential media causes some organisms to take on a different appearance. There's something in there that you can look at it and say, oh, this has changed color, so I know this about that organism. Okay, so one example is using blood agar. You're going to use this again later on if you're in lab. It's actually sheep's blood that we put into the agar. And organisms have characteristic ways that they can break down red blood cells. Okay, some organisms will totally break down red blood cells, lyse them right open. We call that beta hemolysis. Organisms have the right enzymes to do that. Where the media, you can see, turns pretty yellow. All that red color is cleared. You can see right through the medium. Some organisms have enzymes that only partially lyse red blood cells. They actually don't release the hemoglobin, so it gets a little bit lighter, but also a little greenish tone because of that hemoglobin. And then some organisms can't lyse red blood cells at all. We call that gamma hemolysis, and there's really no change in the media for an organism that can't hemolyze red blood cells. Okay, so we have the media, blood agar, and by how an organism changes that media, it gives us some information about how it hemolyzes red blood cells. Okay, we could make blood agar selective as well if we added a couple different antibiotics to it that maybe select against a certain kind of bacteria and allow another one to grow. Okay, so media can be both selective and differential, or it can be one or the other, or something like nutrient agar is neither one. Okay, so the last thing there, anaerobic media. This is just, I don't know, this doesn't really fit here very well in my mind, but this is just media that has some way to get rid of oxygen. All right, some bacteria prefer to live without oxygen or need to live where there's no oxygen. Okay, so one example of an anaerobic media, this is from your book here, but this is one that we're going to inoculate in lab in a little while. It's called thioglycolate media. Basically, it's a reducing medium. So any oxygen that's in that medium gets reduced to water. Okay, when you add electrons, remember, you're usually adding protons as well. Those protons kind of hitch a ride so that oxygen gets reduced to water. So essentially, there's a chemical reaction in that media that gets rid of oxygen. So if we're trying to understand if an organism needs oxygen to metabolize or not, we can inoculate that and then look to see where it grows. If it grows at the top, well, there's a little oxygen diffusing in here under the cap. Okay, there's an organism that needs oxygen to metabolize. If it's only growing in the bottom, then it's one that can't live in the presence of oxygen. Okay, so just an example. There's other physical things that we can do to get rid of oxygen. There's glove boxes and things like that, but that's just an example. Okay, so to finish up talking about culturing techniques, sometimes we tend to think that, oh, whatever I streak out on this media, if I streak the surface of this bench and put it on this media, everything that's on that bench is going to grow on this media. And nothing could be further from the truth. So we know that our culture mediums are not going to grow. Not everything is going to grow on any one media, or there's some things that we've found we don't have a way to grow them in the lab. The only way we found to study these organisms is by maybe growing them in an animal. So for example, leprosy, the organism that causes leprosy, we haven't figured out how to propagate that in the lab. We have to grow it in armadillos. The organism that causes syphilis, Treponema pallidum, we can't get that to grow in the lab either. We have to grow that into rabbits if we want to study that. So we still have a long way to go in culture techniques. And of course, if we want to grow viruses, we have to grow viruses in living cells. So in lab, you streak out a surface, you're not going to get viruses to grow there. Now, we could make a layer, have a layer of bacteria cells growing, and then add viruses and get some viruses that would grow in bacterial cells. But most often if we're growing viruses that would invade animal cells, human cells, we do some kind of container like this, where we have a layer of, say, whatever, animal cells, and then we bathe it in liquid that has nutrients in it and get the viruses to grow up in those animal cells. Difficult is trickier than growing bacterial cells, because this very easily gets contaminated with bacterial cells. So I have to have ways around that. Okay, we think probably 99% of the bacteria that's normally in our body, we haven't figured out a way to culture it yet. Okay, so there's probably a lot that we don't know. So, we have to continue to make advances there. Okay, so that's just a little bit of chapter 6 that we're going to touch on, the culturing techniques. We're going to come back to chapter 6 in another week or two, but there's just I split up that homework assignment so you can do that short assignment now if you like. Okay, so moving on, microscopy. That's kind of the obvious thing that we would be talking about early on here. We're going to think about some general principles of microscopy, meaning what sort of things you have to understand to see the image well. And that's a repeat. You're going to get that in lab as well if you're in lab. We're going to look at some different kinds of microscopy. In lab, you will only do bright field microscopy, but there's a lot of other cool kinds of microscopes out there that we'll just kind of briefly take a look at. I kept this objective here, or this question, what are some principles of staining? Just to let you know that they do talk a lot about staining in your textbook. So if you want a little background from what you talk about in lab, that's a good source. But I'm going to let your lab professor talk more about staining, so we don't have to spend time doing that here. Also included in chapter 4, I don't know, they just didn't want to make 2 little chapters or whatever. They talk about taxonomy. So that at first, at least to me, seems kind of boring, but hopefully I'll convince you that that's important, that we're organized about that. Talk about Carl Linnaeus, about some of things that he started that we've continued today in revising how we think about taxonomy, and some more recent developments that Carl Woese put forward. So that's where we're going. We may almost get through all that today. We'll see. Okay. So first off, a word about units. And I don't, at least to me, sometimes this has been a little bit confusing, but I think we need to be aware of those teeny tiny units that maybe you haven't used to measure anything else. So this is a nice helpful diagram here. So you all know how big a meter is, right? A meter stick, it's about a yard, about 3 feet. Obviously, that's not a very useful unit when we're talking about things that are really small. Okay, we need units ideally that just are a little bit smaller than what we're trying to measure. Okay, so if we measured our microbes in meters, it would be a really cumbersome number, 0.00000, right? So we want to get some smaller units. A millimeter is 0.001 meters. All right, so you move the decimal places the decimal place 3 slots to the left. Okay, and the unaided human eye can see a little bit smaller than a millimeter. Okay, and even maybe the largest protozoans could be almost visible with the human eye. Okay. We really need a unit smaller than that. So we're going to go 3 more places to the left. So this is 0.000001 meters. It's a millionth of a meter, called a micrometer. Okay, so this Greek mu, that letter that kind of looks like a U, that's an abbreviation for millionth. Okay, so a micrometer, or sometimes we call it a micron. Okay, that's a pretty handy unit to measure bacteria, because our typical bacteria and archaea are about a micron, kind of as a general rule. There's some variation, but as a general rule. So human cells maybe about 10 times larger as a general rule than that, about 10 microns across, and then, you know, some of the organelles somewhere in between that. To measure viruses, though, we need something even a little bit smaller, a nanometer. So I guess that's 1 times 10 to the negative 9th meters. So 3 more places over, that's a billionth of a meter. That's a handy unit to be measuring viruses. Ribosomes are about that small, maybe proteins. Okay. So to be seeing that small, you really need an electron microscope here. We're not really going to talk about scanning, tunneling, or atomic force microscopes. Those are a little newer that aren't used as much. We're going to focus on using light microscopy. All right, so light microscopy, we can't see viruses with light microscopy here. Keep that in mind. We would need an electron microscope to see something that small. Okay, so there are a few questions in your homework that ask you to convert between units. As long as you remember that nanometers are smallest, and then micrometers, millimeters, meters, then that will help you think through. It's just a matter of which way to move the decimal place. Okay, so hopefully not too confusing there. All right, so before we get into the different kinds of microscopes, let's think about what we need in order to see our image well. And there's really 3 things that we have to think about, and they're all pretty important. The first thing that you think about is magnification. Well, of course we need good magnification. That's apparent increase in size. Okay, so with this microscope here, this is a bright field microscope, kind of similar to the ones that you're going to be using in lab. You can see that there's actually a series of lenses in there that we call that compound, and there's a series of lenses. But there's lenses here, we call those the ocular lenses, and then there's some objective lenses down here. So you multiply the ocular lens by the objective, and that gives you the total magnification. All right, so with our bright field microscopes, we could magnify up to about 1,000 times. Okay, and actually we could get a light microscope that would magnify more than that. We have lenses that could do that, but we start to get limited by the physical properties of light. Okay, and even if we magnify bigger than that, the edges are going to be fuzzier enough that it's not going to be helpful to actually see what we're trying to see. So we can start to get limited by the resolution. Okay, and we can define resolution. It's a little bit harder to define. The ability to distinguish two objects as separate. Okay. That are close together. Okay, and you're usually talking about when you're looking through some kind of lens. So whether you're looking through a microscope or you're looking through a telescope, okay, we want to be able to resolve that 2 things that are close together are actually separate. Okay, so up in the front here, you can probably tell that my hands are close together, but not quite touching. You can see that there's space in between them. From a distance away, it might just look like I have my hands together. Okay, so when we're measuring resolution, we're actually measuring that distance, that how far apart two things are, and we can still see that they're two different things. Okay, so if your resolution is really good, things are really close together and you can still tell they're separate. If you have poor resolution, things have to be really far apart to see that space in between them. Okay, so with light microscopes, for example, we know that the cell wall is right next to the cell membrane. They look like the same thing. Even though we know that they're separate structures, they look like the same thing. We don't have good enough resolution. We would need an electron microscope to see that they are separate structures. Okay, so if we think about, well, what can we do to make the resolution as good as we can? It's not the same thing as being in focus. You always assume that your microscope is in just right focus, when in reality, that's a little bit tricky sometimes. So if we wanted to kind of improve the resolution, and this formula is in your lab book as well, the resolution is equal to the wavelength of light divided by the numerical aperture. And sometimes there's a constant associated with that equation that we're not going to worry about. Okay, let me define numerical aperture. We start getting into like physics stuff that get like more detail than what I really want to. But the numerical aperture is a measure of the angles of light accepted into the lens. Okay, so we're going to talk about how we can monkey with that a little bit in a minute. But if you think about, okay, I want to get the best resolution that I can, remember, good resolution is going to be a low number. Okay, so resolution or resolving power, it's actually a measure, it's actually a distance. We want it to be really small for it to be good. Okay, so if we want this to equal a low number, okay, this is a fraction. So either we want the numerator to be smaller, right? Because like 1/2, sorry, we want the numerator to be smaller because 1/2 is less than, say, 3/2. Okay, if the numerator is smaller. Or we want the denominator to be larger. Okay, so, right, 1/4 is bigger than 1/5, right? Okay, just a little math review there. Okay, so either we want the wavelength of light to be shorter, okay, or we want to somehow make that numerical aperture bigger. Okay, so how could we shorten the wavelength of light? Well, I haven't actually tried this with our light microscopes, but we could use blue light. Okay, if you remember, if you think about how long light is, like visible light is measured between like 400 and 700 nanometers. Okay, and if you look at a rainbow, like the red's always on top because it's a longer wavelength, blues and purples are always on the bottom because they're shorter. So if we use a shorter wavelength of light, we'll make the numerator smaller, in theory, give us a little better resolution. I haven't actually tried doing that. We just have white light in lab. But if you use electrons, much shorter wavelength. So that's what makes electron microscopes so powerful, because those electrons behave differently than regular light. Another way, so how can we mess with the numerical aperture? The next picture here, I think a similar diagram is also in your lab book. If we were to use a low power objective lens, so you've got several different objectives, you know, low, medium, high power usually. The regular run-of-the-mill one that you've probably used in the past before scanning or low power. So here's the objective at the top here. Here's the light coming up through, hitting your cells. Usually we're not going to have a cover slip on there when we're looking at bacteria, but whatever. If there's a cover slip, that's fine if you're doing a wet mount or something. When the light comes up through the glass and hits the air, okay, it's going to refract. When it changes mediums, it's going to bend. That's just the way light acts. So we lose a lot of light, kind of bends off normally. But if we use a lens that is designed to be used in oil, an oil immersion lens, okay, this is primarily the way we're going to see bacterial cells in the lab. We have an oil immersion lens that's designed to go right down into and touch some oil. So you have to put a drop of oil right on your slide and you turn your lens to go right into that oil. When the light comes up through, the immersion oil has the same refractive index as the glass. So we get much less bending of light and more of those angles actually go up into your lens. Okay, so that actually increases the numerical aperture. I tried to write that out for you there. Okay, so really, we can really barely see bacterial cells unless we use an oil immersion lens. Okay, so to improve resolution, we're going to use oil and an oil immersion lens. A special lens that's only can be focused when you've got it right down in the oil. Okay, if you get oil on those other lenses, like the low power, it just gunks them up. So we want to try not to do that. Okay, again, this will increase that numerical aperture. I'm gonna just abbreviate that a little bit. Okay, so it's a little more complicated thinking about resolution, but again, it's really important to get that as good as we can. The third thing that's really important when we're trying to see our cells is contrast. Okay, that's the ability of cells to stand out from their background. Okay, that's a concept that shouldn't be too foreign. I know back in the day when I used to teach bio 120 lab, I don't know if they're still doing that, but one of the things they used to do is you'd scrape your cheek cell and make a wet mount with that and take a look at it. So these are cheek cells here. And you can see if you just make a wet mount with your cheek cells, it's really hard to see them. Once you find them, then yeah, you can see the edges, but it would be really hard to find those if it was unstained. So one of the things that we do to improve contrast is to stain our cells. Okay, so this picture on the right here, they've added some maybe methylene blue or maybe another dye so that you can see the edges of those cells. So it stands out from the background. Unfortunately, staining cells always kills the cells. Okay. So that's kind of a drawback. You can't see them swimming around, but at least you can see them. And incidentally, all these little dots here near the cheek cell, there's some more inside of it. Those are bacteria. So that gives you kind of a good perspective on how much smaller that generally bacteria are to eukaryotic cells. Here's the nuclei in the middle here. Okay, one other thing that can improve contrast a little bit is to dim the light. If you increase the light, it tends to bleach everything out. But if you're trying to not stain the cells and still see them with a bright field microscope, if you dim the light just right, then that can help a little bit. If you're trying to do a wet mount, then you have to do that to be able to find them. But it's a little trickier. It's easiest just to use some dye. All right, so the types of microscopy that I want to talk about like I said, your book talks about a few other ones beyond this. These are the ones that I want to stick to. I'll give you some examples of what cells look like. So basically you should know about, what's unique about each one of these. Okay, so why is this useful to us to use this kind of microscopy? And to some extent, what do the cells look like? Okay, some kind of, basic idea of the appearance of the cells, or kind of general idea. Okay, so we'll go through these one at a time here. Light microscopy, I assume that if you've used a microscope before, it's probably been a light microscope. The image here is always a light background, unless you stain the background. Sometimes that's you have the ability to do that with certain dyes, but oftentimes we're staining the cells and the background is light. We have ways to stain some cells one color and some cells another color. This happens to be a Gram stain. And I am not worried that you know the parts of a microscope. I'll leave your lab professor if they want to teach you that, that's fine. Remember, we said bright field, they're going to magnify up to about 1,000 times. Okay, and generally, I could have mentioned this before. The resolution is generally about 2 micrometers, 0.2 micrometers. Okay, so, on a good bright field microscope, about 2 micrometers, or you could say 200 nanometers. Okay, if you convert between those units, that's the same thing. Okay. All right, so again, that's what you're probably most familiar with. A dark field microscope, again, this is still using regular light. What's special about a dark field microscope is that it has an obstruction right near the light source, or sometimes we call this a dark field stop. So basically, I don't think they usually have a hole in the middle like this. So basically, there's a disc down here just above the light source, and so here it's kind of turned up like this so you can see it. It would be down flat. So basically, the only light that comes up through your specimen comes at a very oblique angle. Okay, it's not coming straight up, it's kind of coming in from the outside. So because of that, the image is light. And to be honest, I don't totally understand why, like the physics of why that happens. But the background is going to be dark. Okay. And I'll show you an example of that in a minute. The advantage here is that staining is not necessary. And so the advantage to that is that you can see the cells alive. Okay, so cells can be alive. All right, so you can see them moving around. You can sometimes, see if they have flagella to move them all around. Okay. Another light microscope. There's a couple different kinds of phase microscopes. And the newest edition of the book kind of left out this whole explanation of what that means for light to be in phase or out of phase. But it just kind of briefly here, when light rays are in phase, that means the troughs of the waves line up and the crests of the rays line up. Okay, when they're in phase, when the light's out of phase, you've got a crest lining up with a trough. Okay, and when they're out of phase, basically that creates contrast. So if you can get the light to be out of phase kind of on the edges of the specimen, then you're going to be able to see that specimen how it's going to stand out from the background. Okay, so there's what's called a phase plate inside of the microscope to knock those light rays out of phase a little bit better, so that we have good contrast. So good contrast without staining. And once again, because we don't need to stain, we can view cells that are alive. Right, so here's it's a nice picture from your book, just examples of what the same cell would look like with those 4 different kinds, or well, 3, depending how you count it, different kinds of microscopy. All right, so without staining, let's see, without staining, okay, that whatever cheek cell or whatever that it is, it's kind of hard to see with bright field microscopy. And again, see the LM, that stands for light microscopy. So these are all just using regular visible light. Dark field, the cell is a little bit light, the background is dark. Phase contrast, kind of, gradients of gray there. You often see a little kind of halo effect here around cells with phase contrast, just because the light's been manipulated a little bit. There's another kind of phase contrast microscopy called Normarski differential interference microscopy, or differential interference contrast. So in other words, this is also, sometimes we call it a DIC, or Normarski DIC. He must have been the guy that's developed it. It's just another kind of phase contrast microscopy. You can see this image is really kind of nice. It looks kind of 3D. It doesn't have any of that kind of artifact of that halo. What's different about it? It's complicated, but my understanding is that it uses kind of gradients of light rather than, kind of one wavelength or the other. Okay, again, very surface understanding of what's really different about it there, but it's a kind of phase contrast. So you can view things alive. All right, what if we are using something other than visible light? Okay, so with fluorescent microscopy, we're actually using ultraviolet light and fluorescent dyes. So we're using dyes that only fluoresce when you hit them with ultraviolet light. So if visible is between about 400 and 700 nanometers, UV is somewhere, depending on what kind of cells that it's hitting, somewhere between 1, 10, or 100 nanometers. Okay, so shorter wavelength. So remember, shorter wavelength of light, that's going to give us improved resolution. Okay, so shorter wavelength. Okay, still light microscopy, LM. But here's an example of how a really important application of using a fluorescent microscope. For years and years, we've used fluorescent microscopy to help us diagnose when somebody has tuberculosis. It's a difficult disease to diagnose. They take a sputum sample. So that's not just spit, but that's something that's coughed up from the lungs a little bit. And they look at that sputum sample under the microscope. Now, if you just use regular microscopy, light microscopy, it would look like this. Not much to see there. But then if we have dyes that will stick only to a certain protein on the outside of that tuberculosis bacterium. And when you look at that under fluorescent microscopy, you can see those bacterium fluoresce. Okay, so it's a really kind of powerful tool to enable us to see some microbes that otherwise would be hard to see that were there. And fluorescence microscopy we can even add another layer of specificity if we tag that fluorescent dye to an antibody. We're going to talk much more about antibodies later on in the course, but antibodies are so specific. We can make an antibody to stick, to adhere to any protein, basically. We can design them. So for example, if you're trying to see if a sample contains the bacterium that causes bubonic plague, this is Yersinia pestis. You can make an antibody that sticks only to proteins in the cell wall of Yersinia pestis, and if they adhere, they will fluoresce. So we can search a sample and see if the bacterium are there. Okay, so that's just really specific. Okay. And then what's touted as kind of a bridge between light microscopy and electron microscopy, these are confocal scopes. Now we have a confocal scope at Grand Valley. Has anybody been over in Kinchi to see it? I haven't been over there either. Okay, but it looks something like this. This is similar to the model that we have at Grand Valley. Confocal microscopes use ultraviolet lasers as their light source. So it's a very concentrated beam of ultraviolet light. And the computer actually constructs a 3D image. So it looks like there's some ocular lenses here and that you can actually look at your specimen. With any microscope that uses UV light, there's a stop there. So obviously you don't want to be looking into UV light. That would be really dangerous. But also the computer will take sections of the sample and construct a 3D image. So this is some cow medulla cells, I believe. You could actually kind of, with the computer, spin that image around and look at it from all different angles. So it's a powerful tool, not quite as expensive as an electron microscope, and it's not as difficult to prepare specimens and just to keep the whole thing running as what an electron microscope would be. So here's some more examples. I can't remember whether I posted this or not, but the top is just with a regular fluorescent microscope, and the bottom is the confocal. So you can see the first one is it's some brain section. I wrote it down. Human medulla section. So you can see we're getting much better detail, better resolution with the confocal. The middle one is some rabbit muscle fibers. Again, better detail. And the third one is a pollen grain. So, you know, with those concentrated UV lasers, we just get a little bit better resolution there. All right, and how do you tell the difference between a dark field microscope picture and a fluorescent? Well, the dark field is not stained. So if you see, you know, anything colored here, that is probably a fluorescent microscope. But I would agree that might not be easy to know, you know, if your specimen wasn't moving around. Okay. All right, so there's a couple different kinds of electron microscopy. In general, what can we say about electron scopes? Well, I already brought this up. The big advantage to electron microscopy is that electrons have a much shorter wavelength, if you would even call it wavelength, than light. A disadvantage is that cells are prepared and viewed in a vacuum. Sorry, 1C there. Okay. I think. Okay, so a vacuum, that means where there's no air. It turns out that if there's air, when you're trying to, pump electrons at your specimen, the air will scatter the electrons. So everything has to be done in the absence of air, which adds a level of complexity to the process of trying to see your cells. Magnification, generally about 10,000 to 100,000 times. Okay, so on the order of 10 to 100 times better magnification than what we can get with light microscopes. Actually, we can get magnification up to 1,000,000 times. That's just not commonly done. So I just mentioned that because there's a homework question that says, what can magnify up to 1,000,000 times? Okay, it's just not typically done. Okay, and this is a picture down here of a transmission electron microscope. They're pretty big things. They take up some room. You're always going to view your image on a computer screen and not actually looking at it. Okay, so what's the difference between a transmission electron microscope and a scanning electron micrograph or microscope? I'll show you images in a minute, but transmission, think about what that word means, passing through. Okay, here the electrons are going to pass through a very thin section of cells. Okay, so what we actually see is a cross section. Okay, so we see a cross section. So sometimes with the transmission electron micrographs, it's kind of hard to figure out what's what because it's a cross section. Where the scanning is just an image of whatever is viewed. Okay, so basically specimen, when we prepare our cells to view, it's very difficult. I've heard of professors like spending a month trying to prepare their cells just to see them with an transmission electron microscope. What does it involve to prep the specimen? You have to dehydrate the cells. You have to embed them, it's usually a plastic. You have to section it really thin. You use something called an ultra microtome. So picture, with the deli that cuts the meat, only something that cuts it much smaller. An ultra microtome can split red blood cells into 100 or 1,000 sections, 100 slices. Okay, so one cell and 100 slices, that's quite a bit. So you have to slice it up, you have to put it on usually a copper grid, and then of course you have to view it in a vacuum. So it's kind of a long process to actually see whatever you're trying to see, but it does have the best resolution. It's better resolution than a scanning. Okay. With the scanning cells, the prep isn't quite as difficult. The electrons don't pass through, so you don't have to have such a thin section. And the image is going to be a 3D image. It's still not easy to prepare the cells. Usually the cells are coated in some kind of heavy metal like gold. And then of course they're viewed in a vacuum. Okay, here's an example of some images. Okay, these are TEM images, transmission electron micrographs. So like I said, a cross section. Sometimes if you're not familiar with looking at these all the time, that's not immediately obvious, but we can add artificial coloring. There's not really dyes, that's added in later. But both of these, here's the cell wall. This is some kind of capsule outside the cell. Sometimes if you don't know what you're looking for, it's kind of hard to see what you're actually seeing. So that's a drawback. Here, if you just look at the units, 250 nanometers, that tells you that this is not light microscopy, pretty small units. The scanning images are definitely, in my mind, more fun to look at. This is a marine algae, some kind of a diatom. This is some fungal spores. There's a paramecium, some round bacterial cells. So kind of easier to actually see some detail. It's the outside of cells. Okay. Any questions about any kind of microscopy? Okay, there are some different images in your book if you want to kind of look around a little bit. But again, all that we do in lab is the light microscopy. Okay, so taxonomy is a little bit drier. I don't have any big, like, nice fancy pictures to show you here, but taxonomy, let's talk first about what it is. A little misconception about that generally, but why is taxonomy important? Well, we have all this information about organisms that we need to have organized in a way. And we need, if we're talking about a certain kind of cell, we need to make sure that we're identifying that properly so that somebody else studying that same organism in Germany, we need to make sure that we're talking about the same thing. So we need to identify things properly, and we need to kind of, it's important to put things in the appropriate group so that when new organisms are observed, then we kind of say, oh, it has the same characteristics of this group. And then we kind of, you know, can very quickly learn things about it. So what is taxonomy? Well, it's classification, nomenclature, and identification. So classification, that just means putting organisms into groups. Okay, and we base that on shared characteristics. Okay, the kind of characteristics that we use to classify organisms is always changing. We're kind of moving towards more molecular characteristics like sequences of DNA or RNA rather than just how the organism looks. Okay, so it's a constantly changing science, and scientists don't always agree about how to classify something. There are, nomenclature just means that there are rules for naming organisms. And there are some rules that we'll get to, but again, that's something that not everybody agrees on. And by identification, I just mean figuring out what group an organism belongs in. Okay, again, really important to alleviate confusion, make sure that we're identifying it properly, make sure we're talking about the same organism that other people are talking about. Okay, so the guy we mentioned last time, a little history lesson, Carlos Linnaeus, he's kind of the father of taxonomy. And he classified organisms based on shared characteristics. Okay, so the kind of things, you know, at the time that he could easily identify, when he first started classifying organisms, he put all organisms into one of two groups, either they're plants or they're animals. Okay, we've been revising that ever since, but he formed the basis of our modern system, where you might recognize the taxa, kingdom, phylum, class, order, family, genus, and species. Some point along the line, hopefully you learned those. You don't have to have the order of that memorized. For me, King Philip comes over for grape soda. That's how I learned it to remember. We have revised that since, so that I'm not done with that list there. But we spend a lot of time thinking about genus and species. Linnaeus said that, well, how do you define a species? Organisms that interbreed belong in the same species. Now, of course, that doesn't work for bacteria because bacteria just divide. You got one cell that divides into two. They're not breeding. So for bacteria, we have to look at other characteristics, genetic DNA homology or biochemical tests, things like that. So we can't rely on that. It gets a little bit more complicated for bacteria, but Linnaeus was the first one to use binomial nomenclature. Okay, that just means that each organism has two names. Okay, the name of science for an organism is a Latin name that includes the genus and the species. All right, so humans, we're Homo sapiens. No matter what country you're in, we're Homo sapiens. A bacterial example, Escherichia coli. Okay, Escherichia is the genus, coli is the species. We often abbreviate that. E. coli, that's such a common organism that everybody knows what you mean by E. coli. But once you've written Escherichia out once in a paper, then after that you can abbreviate the genus. Okay, so the genus should be capitalized and the species should be lowercase. Okay, just write that in, lowercase. If you're handwriting the genus and species, it should be underlined. Okay, that's the grammatically correct way to write it. If you're typing it out, it's going to be italicized. Okay, and the other rule that he came up with, he said, well, the genus should be a noun and the species should be an adjective. I almost said verb there. Okay, and that's not always obvious and that's not always adhered to, but Escherichia coli was actually named Bacterium coli, and then it was later renamed in the one that discovered it, Theodore Escherich. It was later renamed in his honor. So that's where we took Theodore Escherich, made it sound Latin, added the IE, so Escherichia coli. He named it Bacterium coli, bacterium of the colon. Okay, so it is kind of an adjective, sort of, if you look at it that way. We often pay homage to people that discovered the organisms and put their name when we're naming the organisms. Okay, let's see. All right, so this is I did not post this mostly because I didn't have a good reference for it. I got it out of encyclopedia at the library and it's kind of old, but I just wanted to show you this as further proof that we need to be organized how we're naming things, how we're organizing our information, because, you know, about now, or at least 20 years ago, we knew there was about 4,000 bacteria, 4,000 viruses, but we know that there are many more species out there. We can tell by picking up genetic sequences. There may be as many as 400,000 viruses and 1 million bacteria. So as we describe new species, we have to know like how to group those. Okay, so we'll finish up with taxonomy next time and start getting into bacterial structures. Have a good long weekend. I'll be in my office for a little while if you want to talk to me.